This video shows our work on the American Chestnut Founder Line Transformation Project. Some of these trees in the greenhouse were produced from founder line cultures which were started from seeds of large surviving American chestnuts from different regions of the chestnut range. Our goal is to transform the cultures that made these trees with the OXO gene to produce blight-resistant trees adapted to grow in different parts of the American chestnut range. We start the transformation process with a plate of agrobacterium carrying the OXO gene. Ryan Tull prepares the agrobacterium for transformation by inoculating it into a flask of liquid medium to grow it up to the cell density we need to do the transformation. Ryan places the flask of agrobacterium in a shaker incubator to grow up overnight to get to the cell density we need to do the transformation. The next morning, he takes the flasks out and checks the optical density of the suspension in a spectrophotometer, which will tell us if the suspension is dense enough to use for transformation. Once he knows that the agrobacterium cells are ready, he pours them into a centrifuge tube, and then he spins them down on a centrifuge to make a pellet of the pure agrobacterium. You can see the pellet of the agrobacterium at the bottom of the centrifuge tube. Then he resuspends that pellet in the medium we use for transformation, and he puts that on a shaker while we get the chestnut embryonic cultures ready to inoculate. The cultures Ryan will transform today are derived from a large surviving American chestnut tree in Black Hill, Maryland. Ryan gets these cultures ready for transformation by size fractionating them, that is, pouring them through some nested sieves to get just the size of cell clumps we need for transformation. It takes a lot of rinsing to make sure we separate the different sizes of cell clumps to get what we want. The large cell clumps on the top sieve are discarded, and what remains on the small sieve are the very small cell clumps that we want to use as target materials for transformation. Ryan collects those off of the small sieve and rinses them into a flask with liquid medium, so that the small cell clumps are suspended in the liquid medium. These are the cell clumps we will use for transformation. When we're ready to inoculate these cell clumps with agrobacterium, we collect the cell clumps on a disc of fine nylon mesh. Ryan is pipetting them out, and he uses vacuum to collect the cell clumps on a disc of nylon mesh. And you'll see the vacuum run in just a second. He picks up the nylon mesh with the cell clumps and put it on a plate of gelled medium, because that is where we do the inoculation with the agrobacterium. Ryan retrieves the agrobacterium from the shaker. You can see it's the cloudy suspension there. He unwraps the plate to prepare it for the agrobacterium. And now he uses a pipette to drip the agrobacterium suspension onto the chestnut cell clumps so that the bacterium can infect the chestnut cells and pass the OXO gene into the chestnut cells. These plates with the inoculated cells will incubate overnight. This is what we call the co-cultivation step. This is actually the next morning, and what Ryan is doing is scraping the slurry of chestnut cells and agrobacterium off the plates, and then he shakes the chestnut cells in a wash medium to clean the agrobacterium off of them. The wash medium contains antibiotic that kills off the agrobacterium, but it doesn't hurt the chestnut cells. Then he pipettes the chestnut cells into flasks with selection medium, and the flasks are placed on a shaker. The selection medium contains an antibiotic that stops the growth of the chestnut cells that did not get transformed, while those cells that did get transformed have resistance to the antibiotic, allowing them to grow into the yellow clumps you see through the flask there. The non-transformed cell clumps turn brown and die, while the transformed cell clumps keep growing, and these are the new transgenic events with the OXO gene. To make sure that the OXO gene is really in each of those putative transgenic events, we check for its presence using PCR. Ryan extracts DNA from those cell clumps and prepares the DNA samples for PCR by adding Taq polymerase and the PCR primers that will amplify the OXO gene, allowing us to visualize it in a gel. Here he is loading the thermal cycler to run the PCR, and that will amplify the DNA sequences, the OXO DNA sequences. After the PCR, he pulls those tubes out, and now he's preparing to run the DNA out on a gel. Here, he's loading the DNA into wells at one end of the gel, and you can see the samples there going down into the wells. Then he turns on the current, and the DNA samples run through the gel. Now, you can't see the DNA unless you put it under ultraviolet light, and so we put the gel on an ultraviolet light, and that way we can visualize which lanes have the OXO DNA in it, and you can see that most of the lanes have signal, which means that these are real transgenic events that got the OXO gene. We'll grow those transgenic lines up now in suspension to make somatic embryos, like those on the plate, and we will germinate those or make shoots from them to produce the OXO chestnut trees. These are transgenic chestnut trees here, but they do not have the OXO gene in them. We're still on that step. I also wanted to show you our cryopreservation system for storing the founder lines. Here, Paul Montello is pulling boxes of cryovials out of our liquid nitrogen freezer. Each cryovial contains a copy of a chestnut culture line or transgenic event. Using this system, we can safely store the cultures for as long as we need to and then thaw and regrow them as needed. I hope you've enjoyed seeing the process we are using to transform the founder lines with the OXO gene to produce blight-resistant chestnuts adapted for growth in different regions. We invite you to visit our lab in the Warnell School at UGA to see it in person.